So welcome to a seminar. So this is a seminar which is run by the Water Security Alliance, which is across the GW4, so it's Bristol, Bath, Cardiff and Exeter. And these run every Thursday at 1 p.m. And this year, well, this term, they're run by fresh CDT PhD students, which I am a member of. So a little bit about me. So I'm a third year PhD student based here at the ESI down in Cornwall. And I'm funded by NERC Fresh CDT, so that's a freshwater training body. And I'm supervised by Professor Gav Iwandurosha, who sadly can't be here today. And my project is looking at how multiple stressors and biodiversity loss together will affect microbial communities. So hopefully James's talk today will be great interest to me. And so I'm joined today by James Orr, who is from the University of Oxford, doing a postdoc there with Michelle Jackson. Yeah, and so you previously did a PhD at Trinity College Dublin, where you studied freshwater ecology and multiple stressor theory. Yeah?
Yeah.
Perfect. Okay, so I'll hand it over to you, James.
Okay, perfect. Yeah.
Perfect.
Do you think this is
If
working?
it's not working, I can...
No?
No?
I can do this.
Yeah, should I stop?
Yeah, sure. It's okay if I stand here and click this with you all? Yeah. Okay, well, hello everyone. Yeah, I'm James. Really nice to meet you all. It's a real pleasure to get to do an in-person seminar after so long. And it's a great excuse to come down to Cornwall. I've never been before. So yeah, I'm working with Michelle Jackson, and we're very much interested in multi-stressors and their interactions. And yeah, that title there, maybe overcoming is not the right word, or maybe I should have a question mark at the end because that's a bit too ambitious. It's more like facing or confronting the context dependency and what we can do about it. And I hope that I'm going to persuade you that maybe we can use more theoretical approaches instead of just many experiments. Maybe we need to take a little step back and have a little bit more theory in terms of the mathematics and the basic biology behind these stressor interactions. So I'll just start by kind of convincing you that multi-stressors are everywhere in every single ecosystem. So this beautiful landscape was very near my fieldwork site in New Zealand, and it looks very pristine and picturesque and untouched, but in reality, it's really devastated by multi-stressors. So being in New Zealand, there's going to be many different invasive species in the landscape. Some of these are completely overgrazing the grassland up there, which didn't used to be grassland, it used to be forest, but it was chopped down. Maybe further down in the valley there, I don't know if you can see that, but it says pesticides. There's lots of farming and agriculture going on, releasing pesticides and microplastics into the environment. Various different sources of sedimentation seeping into the freshwaters as well. On land and in the water, there's increased nutrients. Maybe there's salinization of the freshwaters from how the roads are protected from ice. Further up in the valley, there's heavy mining. There's mining going on, releasing metals into the environment. And all of these local stressors that are impacting the environment are also interacting with these global stressors as well. So it changes to temperature and rainfall patterns, all of these different environmental factors being changed by human activities. That's what we call a stressor, really. And all of these stressors, of course, are interacting in this really complex web, and they change each other's values, they change each other's effects. And a lot of the time we focus on one or two stressors at a time and make a factorial design, but in reality, there's many different stressors. So, and of course, there are many stressors we don't know about yet, or that are still emerging, novel pesticides, new invasions, and so on. So these novel stressors are going to interact with the current web of stressors that we already have. So it seems a bit crazy, and what can we do about this? But I'm going to say maybe we should take a step back, and instead of building up all these different predictions using different experiments, we need to take a step back and get some really good kind of a theoretical framework for how we can understand stressor interactions. So in the talk, I'll kind of explain what I mean by this context dependency. And then I'm going to present three kind of pieces of research. I'll kind of fly through them. And they're research that I did some during my PhD and some in the past year at Oxford. And they're all about kind of, they're all like theoretical approaches to try and generalize stressor interactions. So instead of just being specific to a certain pair of stressors in a certain system, these kind of mechanisms and approaches are general to huge collections of stressors. So that's what I'm going to focus on today. Okay, so but just to begin, in case you're not familiar with the stressor interaction stuff, let's say we've got two stressors, A and B, and we measure their individual effects on some biological response. The question that most multi-stressor people are interested in, what's their combined effect going to be? When we put them together, what's going to happen? And so we make some prediction. This is the knowledge that we start with, and then we make some prediction. And a very classic prediction would be just to add the effects up. So that's the additive null model. Or it's kind of linearity and all these kind of ideas where the two effects are independent of each other. We can just add them up. And so maybe we then do a multi-stressor experiment and we have a factorial design and we can actually measure what they are together. And there's kind of three possibilities. Either, great, our prediction was right, in which case we say there's no stressor interaction, but nearly all the time there's going to be one of two other scenarios. And they are, that's what we call stressor interactions. So when the combined effect is less than what we predicted, that would be termed antagonism. So it doesn't mean that the effects are getting reversed or anything. It just means that it's not as bad as we expected. So maybe the response here could be the number of species that went extinct. And if it was one stressor caused 5 extinctions, another one caused 4 extinctions, together we thought they might cause 9, but they only caused 8. That's antagonistic. But another perspective and another very common scenario would be this synergistic outcome. And I don't know if in the room we can see it, but the bar is higher than the prediction. So when the combined effect is greater than what we expected, that's a synergistic stressor interaction. And statistically, this is like an interactive term in an ANOVA or something like that. Okay, so that's what I mean by stressor interactions. I'm not really talking about like two stressors in the wild influencing each other's like in a direct way that could cause this. But what I'm kind of referring to is the statistical outcome from like a multi-stressor experiment. Cool. So that's our baseline there. And we do some experiment with A and B, and some policymakers might want to say, okay, well, what happens when you put A and B together? Should I be worried? Are they going to be synergistic or antagonistic? And that would be the goal of doing all this multi-stressor research, is to have recommendations for policymakers to say, oh, when these two stressors are together, it's a bit of a disaster. If you get rid of one of them, it'll have a huge impact. Or you might find that when they're together, they have the same effect as just one of them. So you could put loads of money into removing one of the stressors, but it doesn't really change the combined effect. So that's kind of the motivation behind understanding these interactions. And so it would be really nice if we could say we could take two stressors and they always interact in the same way. But that is not the case at all. There are so many different things that their interaction depends on. So it depends on what prediction I made. So I could have used that additive model, but I could have used any model, a multiplicative one or some more complex one. In ecotoxicology, they've got loads of different models. And it's not always totally clear from the statistics that people use which model they're using. So that can change how we interpret the interaction. There's also a bunch of classification systems. So I've just said there's antagonistic and synergistic, but if you read the literature, there's so many other words out there for these things. There's reversals and mitigation and amplifying and dampening and different research communities sometimes mean different things by these. So it can be quite hard in meta-analyses to pull together information. Also, because we're like statistically measuring these things, it matters how many samples we're taking. And if we do two identical experiments, have the same effect sizes, but if one experiment had 4 replicates and another had 10, one might say, oh, these are additive, but the other could say it's synergistic. So we need to look at the effect sizes as well. It also matters how long we're looking. Biological responses change over time for things like evolution. Or what scale are we looking at a lab in a little microcosm, or at a mesocosm, or in a river, or in a whole catchment? The stressors can change how they're interacting. What are we measuring? What was that response? Is it species loss? Is it total biomass? Is it certain genes in an organism, in a population? All these things completely change how the stressors statistically interact. And And if we're looking at the higher levels, like communities, it matters which species are there. Some species can mitigate the effects of stressors. They can amplify it. The effect of stressors can get propagated through a web of species interactions. It matters if the stresses happen far apart together, if they're ramped, if they're constant. So there's a lot going on. It matters how strong they are, and it matters on other stresses as well. So we have a third stressor in here. There could be a higher order interaction. In the same way that we could think of three species. Two species may interact when they're together, but when there's a third one, it may cause some other type of interaction. So really, when we want to say how do stressors A and B interact, that's what I mean. It depends, and that's what I mean by this context dependency. And yeah, what can we really do about this? So in our lab group, we're doing like a little synthesis at the moment, and we just looked for multi-stressor experiments that are in freshwater systems, and we're found close to 3,000 different experiments, which I think is that's a huge number. Like 3,000 different experiments have been done, and when you look at meta-analyses, there are not a lot of general rules and patterns. We can't say A and B should interact in a certain way. So there's something that we're missing here. Like these meta-analyses and meta research in general is obviously, it's not moving. Might just catch up in a second. Oh, there we go. Yeah, because there are so many different experiments and there's a lot of context dependency, meta-analyses have been kind of the primary tool that we use. And there are all sorts of problems with doing that. But yeah, they're useful and they point us towards potential problems and they highlight context dependencies. But I would argue that we really need to strengthen these theoretical foundations. So we need to really take a step back and think of stressors abstractly and think of what's the mathematics going on behind these interactions and how can we generalize to all stressors, to any system. So I'm going to present some research. Some of it's verbal theory and some's mathematical theory, but it's all this idea of just like taking a step back and not focusing too much on the identity of stressors, but just thinking of abstract stressors and abstract interactions. Okay. So first example of this kind of approach that I'm talking about is considering trade-offs in adaptation. So what I mean by this is, say you have a population that is exposed to multiple stressors. Over time, they're going to be adapting to these stressors through all sorts of different mechanisms. So there's like plastic responses, if they're small, like microbes in a short time from our perspective, they can be evolving. And so they're going to have to perform multiple tasks. So maybe the two stressors are warming and some pesticide. And they've got two tasks they've got to do. They have to adapt to the warming and they have to adapt to the pesticides. And they have to do it at the same time. And so when organisms have to do multiple things at the same time is typically a trade-off. And so we kind of say, oh, how does this trade-off influence stressor interactions? Because trade-offs are such a general thing in biology. So on the left-hand side here is just another way of showing what I mean by a stressor interaction. So instead of a bar chart like I showed at the beginning, I'm just going to show that this phase space here, it's not really phase space, but this plot here, on the X and Y axis, you've got the effects of the individual stressors. And on the Z axis, you've got this way of representing additivity. So if you go straight up from the X and straight across from the Y, that's where the additive expectation is. And so the combined effect of the two stressors here is additive, just for simplicity. But the combined effect could be less than we predicted, so antagonistic, or it could be more than we predicted, synergistic. But for now, let's just say the prediction was spot on at the beginning. And so that's this framework of stressor interactions on one side. And then in terms of trade-offs, we can plot a different type of chart here. So this is performance space. And on each axis, it's the adaptation to one of the stressors. And so a population can fall anywhere in this space. It's not confined to the lines like on the left-hand side. And so this is the baseline. This is the populations have been exposed to the stressors and we've just observed the initial effect. So of course, there's no adaptation yet. It's like time zero. But if we watch the system for a while and the populations are adapting, then that's what I'm going to represent these red points by. So on the left-hand side, you see that after adaptation has occurred, the effects of the stressors have all gone down, which makes sense. That's what adaptation is. So the effect of the stressors have gone down. And on the right-hand side there, you see that adaptation has gone up. So it's just the two ways of showing the same thing. And what's happening here, importantly, is that the distance along the diagonal is equal to the sum of the distance along the vertical and horizontal. So what I mean by that is that this adaptation to multiple stressors is equal to the sum of the adaptation to the single stressors. So it's as if the adaptation was independent. It was just able to adapt to temperature and adapt to the pesticides. And when they were together, it just put its adaptation together. And so when this happens, you've got this little equality here. And any interaction before in the blue will be preserved to the red. So the stressor interaction won't change. So if you've got this equality down there in terms of its adaptation, then stressor interactions won't change. But I could see a few faces kind of looking a bit surprised when I said the sum of adaptation would be equal to the adaptation to multi-stressors. And that's because of trade-offs. It's not going to work out like that. So on this other side here, I've just put in this thing called the Pareto front. So it's this idea in I think it came from economics originally, but it's quite popular in evolutionary biology. And it's just that there's a certain space in performance space that is unreachable. I just can't get there. There's a trade-off. Maybe we'll take a step back, and instead of thinking of adaptation to stressors, you could say that the organism is being asked to fly and to run. And it's never going to be a perfect flyer and a perfect runner at the same time. You can't have wings and be a sprinter. You got to choose one of the strategies. And so that's the same thing going on here. So if you're adapting to temperature, you get to some boundary. And if you want to adapt any further to that temperature, you have to sacrifice your adaptation to pesticides. So that's what we mean by if there's a trade-off, this will happen. And so you might be able to see it already, but if there's a trade-off, then there's not going to be that equal along the diagonal. There's this inequality. So it's the adaptation to multi-stressors is less than the adaptation to the sum of the stressors. So that translated over here, that's what this means. And you might be able to see already that the blue points are going to be additive and the red points are now synergistic. So even though the combined effect is less than it was before, it hasn't reduced as much as we expected it to. So if you just focus on the blue points, it's still additive. But if you focus on the red points, it's a qualitatively different scenario now. It's synergistic. So the conclusions about what we should tell policymakers about these stressors has changed dramatically. And we now say, oh, if you remove one of these stressors, it'll have a big effect. You know, it's kind of a strange scenario that these stressors can just change due to something like evolution. But the underlying mechanism is something very general, just a trade-off. So yeah, this inequality here will be the bias towards synergism. The greater the inequality, the bigger the bias towards synergism. Okay, so that's all nice. But does it actually happen with real systems? And so we took data from an evolution experiment with rotifers. And these rotifers had been exposed to multi-stressors for about 70 days or so, which is about 30 generations. So it's a lot of time for rapid evolution to occur. And yeah, real data is always a bit messier than my little plots. But you'll see, I want you to just focus on these inequalities. So the three stressors were salt, copper, and temperature. And every combination of the stressors, we saw this exact same pattern. So we're seeing all these inequalities here. So in all cases, there's a shift towards synergism. And in all cases, there's evidence of performance trade-offs. So it seems like it could be a bit of a general mechanism of synergism in natural systems, because organisms are facing trade-offs when there are multiple stressors. So yeah, that's the idea there. I'm very happy to answer any questions or chat about the evolution experiment after, but I think I might just like plow on through for now. So yeah, so just some closing thoughts on this kind of idea of the importance of trade-offs. I was just showing like it in 2D, so just two stressors, but obviously at the beginning I was alluding to this idea that there are many, many different stressors. So when we add more stressors in, the dimensions of performance space increases. And so does that increase the size of the inaccessible area? when you have more and more stressors, are there more and more trade-offs? And if that's happening, then there might be a greater shift towards synergism. That's just an idea. We also had a 3 stressor scenario, and I have a little video of it, and it does have a bigger bias towards synergism. But yeah, just something to think about. And then this other idea in this multi-stressor community, we talk about like modes of action a lot. So if stressors have a similar mode of action, maybe we can just put them together. So if they both do the affect an organism in the same way, it's very easy to put them onto the same kind of scale. And if they have very similar modes of action, then maybe there's not going to be much of a trade-off. Adapting to one stressor may help you adapt to the other stressor. There's these kind of pleiotropic effects. And so really this kind of idea of a shift towards synergism makes most sense if you think of very dissimilar stressors. But very similar stressors, you may not find it. I don't think you would. Cool. And then actually touching on what your experiment's going to be, Hebe, is like this idea of what do the species interactions do. So I'm just kind of thinking no species interactions and just single populations. But as soon as we put species interactions, how does this change how evolution plays out with the multi-stressors? So I think that's probably the next step with this kind of research. Cool. And so if you're interested, it was published in Global Change Biology, this stuff, with Jeremy Piggott, Pepijn Leutz, Andrew Jackson, and Jeff Arnaldi. And so there's like a mathematician, an evolutionary biologist, a modeler, and like a freshwater person there. So it was like the paper has a lot of different perspectives in it. But hopefully it makes sense. But yeah, I'm very happy to chat about that later on if you like. Cool. So yeah, I'm going to move on to this next topic, which is the observation of the community. And this is a bit more in my area of expertise. I'm like a community ecologist. So the evolution stuff was a bit daunting to me, but this makes more sense. And it's a general approach that we're going to be taking. So I'm going to be talking about abstract stressors, A and B and stuff. I'm not going to be identifying what they are. But anyway, what do I mean by this? Okay, so let's say that you've got a bunch of freshwater mesocosms and you've got 2 stressors and you want to know how they interact. And you apply the stressors individually and combined, and you look at changes to the zooplankton and the macroinvertebrates and the phytoplankton and everything. And you build up, you get all your data, and then you say, okay, well, how am I going to measure the effect of the stressors? And you might say, okay, I'm gonna use 2 response variables. I'm going to look at the changes in total biomass, and I'm going to look at the changes in the diversity. So maybe like species richness or Shannon's index or one of those things. And you say, I'm going to take these 2 response variables, and I'm going to report both of them in my paper. That's like a priori. That's what you decide to do. And it would be nice from a multi-stressor perspective if they both observe the same stressor interaction. So on this plot here on the x-axis, I'm showing like the stressor interaction of biomass, so like non-additivity of biomass, and on the y-axis is the non-additivity of diversity. And so if there was no stressor interactions, the point would be bang in the middle. But if there are stressor interactions and both response variables observe antagonism, it'll be in the bottom left. If they both observe synergism, it's the top right. And if there's a mismatch, which is not what we want, that's like this context dependency. Response variables aren't telling us the same thing. Which one should we choose? Which should I report? They have very different implications. And there's already evidence that there's a publication bias towards synergism. Editors are more likely to publish papers that report synergism compared to antagonism. So maybe you're more likely to report the one that showed synergism. So that's not great. So we then said, okay, that's the problem that we've got here. Is this a real problem? And so we plotted some data from meta-analyses. So Michelle Jackson has a great meta-analysis from all this kind of stuff. And we took all the experiments that looked at 2 stressors, but they used biomass and they used diversity at the same time. And we thought, okay, most of them will be in the blue, and there might be a couple in the red for various reasons. But actually, we found this kind of systematic bias, like it's more likely that you're in a mismatch here. And we've since done it for more experiments, and we're finding this kind of thing, that when you have biomass and diversity, there's actually a greater than 0 chance that you're going to be in this mismatch zone. So what does that mean, and how can we explain this? So so the 2 things that are at play here, there's the observations that you're making of the community, and there's also the dynamics of the community. And what's nice is that these 2 are linked really together through mismatches of stressor interactions. So what I mean by that is the consistency, like how if we observe a mismatch or a consistent pair of responses, it depends on how collinear the observations are, so how similar they are, and how consistently the species are responding. So by collinear responses, I'm talking about like linear functions. So let's imagine that this is our system here. It's just got two species, and it's the phase space here. We'll just plot the state of the community is going to be the biomass of the two species. So it can only be in the top right-hand corner because we can't get negatives of the species. So let's say that the two species are a mealworm and a mushroom, just for sake of argument now, and that the state, the initial state, is up here. So we've got a certain amount of mealworms and a certain amount of mushrooms, and that's, we can describe the system like that. But if we wanted to give a single number instead of representing its like composition in a matrix or yeah, if we wanted like a single community aggregate property, we need to project onto some function. So total biomass is the classic one because it's just summing up all of the biomasses. So it's this diagonal one that's a bit similar to the trade-off plot I was showing. But this is what total biomass looks like. It's just a straight line along the diagonal of phase space. And we're just adding up all the biomasses. So you project the state onto the function there, and that's what your total biomass is going to be. But another function that you could think of might be decomposition. And maybe in this system, the mushrooms are slightly better at breaking down general stuff than the mealworms. So that function is going to be tilted slightly towards the mushrooms. But another function, especially when you're talking about mealworms, is plastic. So mealworms are great at breaking down plastic. So this function here for this system might be tilted really in favor of the mealworms. So I can change the mushroom biomass as much as I want, but it's not really influencing the ecosystem property of plastic decomposition. But if I change the mealworms at all, it's going to be changing the plastic decomposition. So this is like this multifunctionality view, and they're kind of linear functions here. Okay, so now instead of biomasses, we're just going to consider non-additive terms from a stressor, from a multi-stressor experiment. So we've run some multi-stressor experiment. We know that we've made a prediction about the combined effects. We know the actual combined effects and the difference between those. That's what we're going to plot for each species here. So that's the non-additive term for each species. Cool. So yeah, if the now points don't just fall in that top right-hand corner, because you can get positive non-additivity and you can get negative non-additivity. So the negative non-additivity is antagonism and the positive is synergism. So yeah, if the point now falls in the top right-hand corner, it means that both species responded synergistically to the stressors. And in the bottom left, both responded antagonistically. And so that's kind of like this compositional view. But if we want to get a functional view and like we observe the system, we do the same thing. We project the non-additivity onto the function. So if our non-additivity vector was up here, we project it onto the function. And if it's, you know, up in the positives, it's going to be synergistic. If it's in the negatives, it's going to be antagonistic. And really what we can do is we can draw this dashed line going through the origin perpendicular to our function. And anything to the right-hand side of this will be observed as synergistic by total biomass. And anything to the left-hand side will be observed as antagonistic. So we can do the same thing for plastic, where we take a dashed line going through the origin perpendicular to the function. And that's what this looks like. And again, anything to the right-hand side is going to be synergistic and anything to the left is antagonistic. And our question is, when do they both see the same thing, or when is there a mismatch? You might be able to start to see it already, but really anything in these blue zones, if you're to the right of both or to the left of both, that's when you observe the same thing, that your observations are consistent. So if the non-additivity vector was up here, both functions are going to report synergism. And down here, both will report antagonism. But in here, you can see when I project onto plastic, it's synergistic, but when I project onto total biomass, it's antagonistic. So that's the problematic cases that I was talking about. Like, we don't like these. This is a problem for multi-stressor research. So really anything in these red zones is not great. And what's nice is that this angle here, so the angle between the two functions, so when I was talking about their collinearity, the angle between the two functions is a really good predictor of the size of these red zones. So when the angle is very small, like between decomposition and total biomass, the red zones are going to be very small. So the potential mismatches are going to be low. But when it's very large, so I've added another function here, like the production of some chemical by the mushrooms, but that's not done by the mealworms. So we've got a very large angle between the functions. And so therefore we've got a very large red zone of mismatches. Okay. And what's cool, because these are orthogonal lines here, that that angle down there in the red zone is the same as the angle between the functions. And of course, it's like we're measuring these in radians. So there's going to be, 2 pi radians in a full circle. And we can get 2 of these thetas. So I'm just going to cancel out those 2's. And I'm left with theta, the angle between the stressors, divided by pi, so 3.14. And that will predict how many mismatches we see, which is kind of nice that a number like pi can appear in a community ecology problem. Cool. So say we run lots of simulations to test this. So first step is you generate some community. So it could be Lotka Voltaire, it could be something very complex, or it could be something very simple. It really doesn't matter. You generate your community, you generate 2 functions, and then you run many multi-stressor experiments, and you get many non-additivity vectors. And for those functions, you can calculate how many times they're agreeing and how many times they're disagreeing. And so this prediction that we have is that it's the angle between the functions divided by pi that should be able to predict the proportion of mismatches that we observe. And yeah, it's it does that. if the slide changes. Yeah, there we go. So it does work. Like when we set the problem up in a simple way, we can demonstrate it very quickly. When we put species interactions in, it doesn't change things. It's just like a nice geometric effect that emerges from making linear observations of systems. So that's kind of cool. But I also said it depended on the consistency of species responses. And so what I mean by that is, in that example before, I was randomly sampling the non-additivity in a unit vector, so anywhere in this space. But maybe there's some reason why that's not the case. Maybe the species are responding in a consistent way for some reason. And there could be biological reasons for that. But yeah, if that's the case, you might notice that the inconsistent species responses are exactly where those mismatches were. And that's just by chance. That for linear functions, the mismatches are going to be in these offsets, which is also where the species are inconsistently responding. And so, yeah, when you overlay the 2, you see that the reds are lining up and the blues are lining up. So when we have more consistency, we're going to expect more blue than chance. So what we would expect then is, I'm going to color the points based on how consistently the species are responding. And they do kind of collapse away from our prediction, but in a very predictable way. So now you've got these 3 ingredients. So you've got the angle between the stressors on the x-axis. You've got the proportion of mismatches on the y-axis, which you can get from doing multiple observations and multiple different stressors. And then on the color is now how consistently the species are responding. And if we know 2 of these, we can figure out the 3rd. And we've got like the equations to do that. So it's quite cool. Like we can take a step back and look at systems from the top down, make lots of observations, and the number of mismatches we see can tell us about the underlying dynamics, the underlying response to the stressors. Or we could start from the bottom, look at all the different the species responses to the stressors, and then we can say, well, total biomass and diversity should mismatch in this case. So it's quite cool. We can come from both angles. Yeah. So these were linear functions. Like I was just drawing lines onto the face space, but a lot of the functions that community ecologists look at aren't linear. So things like diversity, very nonlinear functions. But what's cool is that there's still a notion of an angle there, but the angle changes based on how even the community is. So it's it takes a bit of while to like draw it out and chat about it, but you'll just have to take my word for it. But even for nonlinear functions, we still get a perfectly good prediction. So it's easiest to explain with linear functions, but they don't need to be linear for this to happen. So something like the Shannon's index or species richness or these kind of things, we can still make these plots. And what you'll notice before for the linear functions, they were kind of confined to about 0.5 mismatches. So that would be just getting like a 50-50 mismatch and consistent responses. But when you add this nonlinearity, we can see that it's going all the way up to 1. So that's when we saw for the empirical data at the beginning that there's actually more mismatches than 50-50. So for biomass and diversity, what we saw in the empirical data anyway is some points up near the top of this plot where you should expect them to mismatch. Okay, cool. So they were the two kind of in-group pieces of the puzzle for this kind of research. And what's nice is that, if you have two community properties, sometimes you should just expect them to observe different things, and that's fine. And you shouldn't worry too much if they do observe different things, and you should definitely report them both. And maybe it's actually hard from a community level to make any inferences about how the stressors are actually interacting, like chemically and physically with each other, because there's so much things going on in between. By the time we get up to the very top, there's just so many more non-additive and non-linear responses that are thrown in. And even just using diversity to measure a stressor interaction, that doesn't make sense because it's a non-linear function. So you're adding non-linearity in just purely by observing it. There could have been no stressor interactions or no species interactions or nothing going on in the system. But if you chose diversity because it's a non-linear function, you will see it like a non-additive response between the stressors, which I think is kind of funny. Okay, so we haven't published this yet, but we'll probably put it up as a preprint pretty soon. But it's similar people, but now Michelle was working with us on this as well. So I'm just gonna briefly for like the last, I'll shoot through this one for like 5 minutes. Is that okay? This is a bit different now. So the other two were this idea of like, you got like some stressors and we want to understand the mechanisms of their interactions and can we generalize? And that's a bit of like a reductionist approach for multi-stressors. So if you build up putting the stressors together, but another way of coming at this problem could be from like the top down, like a holistic approach. You gather a whole pool of stressors and can we use aggregate properties from these stressors to say anything about their combined effect? So that it would be like an analogous to like the biodiversity ecosystem function. It might be like stressor diversity ecosystem collapse or something like that, you know? So instead of building up all these pieces of the puzzle like I've been doing so far, this idea is kind of getting at the other approach. Cool, so I like this little poem here. So I keep 6 honest serving men. They taught me all I knew. Their names are what and why and when and how and where and who. And we applied this to the question of, are stressors similar to each other? Because we need to figure out if they're similar so that we can, start to describe a whole pool of them. So what's nice though is that these 6 questions, they pop up in journalism and philosophy all the time, but they are also, there's actually 6 kind of concepts that in multi-stressor communities where these are directly applicable. So like, what are the stressors? Why are the stressors present? When do the stresses occur? Where do they occur? How do they affect organisms? And who do they impact? And each of these 6 points, there's research that goes on in that. And stresses are compared based on these kind of questions. So let's say we've got 3 stressors, A, B, and C here, and they impact like biological systems in different ways. They can enter at lower levels of organization or at the top. They can directly interact with each other, or they can interact with their effects. All sorts of weird things can go on. And we look at these 3 stresses and we say, well, are they similar to each other or not? That's the problem. And we could say, well, what are they? Are they 2 pesticides, or are they a heat wave and something completely different? You look at their traits. That's one approach, and that's very commonly done. You could look at why they're there. So that's this idea of like, is there like a similar pathway? So can we deal with them in the same way? Are they both from anthropogenic climate change? Are they both from agriculture? Are they from a mix? These kind of things. So that's like the red list, the threats on the red list does it that way, and a bunch of other systems. Then there's this idea of when do they occur? When do they occur? So do they occur simultaneously, or are they lagged? Are they like waves? Are they ramped? Are they pulsed? We can match up their timings and see how similar their timings are as well. We can obviously match them up on a map, see do they occur in the same places, in the same ecosystems. And then I like this one because it's on communities. It's the idea of who do they impact in a community. So it's called Cotolerance. and it's probably like some of the earliest multi-stressor theory. It was done in like 2004 or something, but it's where you plot on the axes the effective stressors, and you plot the species there. And if the species are responding in the same way to the stressors, they're going to be correlated in this space. So you'll get basically a single number. The correlation between these points can tell you something about how similar the stressors are from a community ecology perspective. So that's my favorite one. But there's also this mode of action one that I mentioned before, and that's done if you're looking at populations and stuff like, does it influence their reproduction or their feeding or stuff like that? So basically, there's a lot of different ways of answering the question. So maybe you can think of an example. There's some lake, and there's every time there's a lot of rain, pesticides come into this lake from the nearby agricultural land. But also a few years ago, some invasive fish that eats insects, an insectivorous invasive fish was introduced to the lake by accident. So this is like a classic scenario. We've got these two stressors, and you might want to say, well, how similar are they? Well, based on the 6 kind of topics that I had before, it depends. So their sources are completely different, their traits are completely different, but they occur in the same place around the same time, and they also have the same cotolerance. They're both influencing insects, the insecticide and the insectivorous fish. Although they seem very different at first, maybe their effects are going to be the exact same. But their modes of action could be completely different. So that was like this kind of framework that we kind of just like set up in a little perspective piece. But the whole point of it was to kind of get to these questions like what's this idea of stressor diversity? So it's like this holistic approach to a big pool of stressors. Is there some way that we can use this aggregate property to predict another aggregate property? So maybe you could picture like 5 very dissimilar stressors compared to 5 very similar stressors, and you'd probably expect the dissimilar ones to have a bigger impact. So their diversity is higher, they're impacting more components in the system instead of the redundant stressors here, like 5 like very similar pesticides, they kill the same pests, that's it, they're done. Whereas 5, a heat wave, a pesticide, a herbicide, a fungicide, and an invasive species, that could really transform the system. I'm not saying that pesticides wouldn't, but you could have a bigger impact if you got a greater diversity of the stressors. So that's one idea. And then there's another thing it's a shame you can't really see it there, but these are stressor response curves. So where we increase the intensity of the stressor, and then we look at the response. So is it linear or is it some non-linear shape? And if 2 stressors have a similar stressor response curve, we can put them into the same language and the same currency, and we can build really good predictive models. So that's also this kind of motivation behind this. It's done a lot for populations because of the mode of action concept, but maybe we can look at other levels, like at a community, we could maybe just put in the cotolerance, and at a whole kind of catchment scale, you could look at spatial temporal overlaps to get a similarity number. So that's that kind of idea. It's not super well developed, but I think it's cool to think of like stressors as like species, and like in community ecology, we sometimes do it from a reductionist approach where we build up all these models from all the species and then try and predict something at the end, or we do something like biodiversity ecosystem function or something like that. And I think we could maybe potentially do the same for the stressors. But yeah, is there a risk of overcomplicating things? I've said there's so many different stressors with so many interactions and higher order interactions, but maybe there's just 1 or 2 really dominant stressors. Maybe if we actually just got rid of nutrients and pesticides in rivers, we might not need to worry about too much other things. That's just, there'd just be a slight warning there for overcomplicating things, maybe. But yeah, so that's this idea of stressor similarity. I think I'm kind of close to time, so I'll thank Matthias and Jackson, who wrote this perspective together. It's just come out in Natural Sciences. And yeah, it's just a short little piece, but I think it's kind of nice to chat about. Cool, and I'll just close very quickly that in our lab in Oxford, we've got loads of theories, but we're also testing them. People who are better with real things. We've got like some PhD students and postdocs working with microcosms. We've got like pond mesocosms that we've actually started wrapping like the insulation you get in camper vans around the edges, and you're seeing like a +2 increase of warming. So this passive warming is kind of cool. That's something we're working on. And we've also got these river mesocosms. I don't know if you might not have heard of it, but we like pump water out of the river directly through these little circular channels. So yeah, we've got like different mesocosm setups, and we're going to be testing some theories that we make about these stressor interactions. Yeah, so I guess I'll just thank you all very much for listening. These are the people that I worked with over here. You can't see Dr. Pepijn-Leutz, but he was the guy who did the rotifer evolution experiment. And yeah, thank you all very much for listening, and I'm very happy to answer any questions.